So dear students, today we are going to discuss about the topic multi-enzyme systems, and within this topic we will focus about their occurrence and properties of multi-enzyme systems. So before we exactly come about to study about the properties of multi-enzyme systems, let us first understand how does this concept of multi-enzyme system came into existence. So for most of the metabolic pathways, a number of enzymes catalyze the sequence of reactions linked such that the product of one enzyme catalyzed reaction becomes the substrate for the next enzyme. For instance, if there is a metabolic pathway where your initial substrate is A and your final product is F, so then the conversion of A to F is occurring via a series of steps of catalytic transformation, and for every step of reaction there is a particular enzyme which is participating. Further, these transformation are following a particular sequence, which means that initially when enzyme E1 will act upon A, then only B will be formed, and E2 is specific only for B. Likewise, E3 is specific for C only, E4 for D, and similarly E5 for E, which also suggests that the product of one step of reaction becomes the substrate for the next step of reaction, or the product of one step of the reaction becomes the intermediary substrate for the next enzyme or the next catalytic center. So throughout this discussion I'll be using these terms: catalytic centers and intermediary substrates. So in the context of this type of representation, A would be your initial substrate, F would be your final product, whereas B, C, D and E, they are called as intermediary substrates, and your enzymes E1, E2, E3, E4, E5 are the participating enzymes of the multi-enzyme complexes, or we call them as different catalytic centers. So, for such a situation, if A is the starting substrate and F is the end product, then the efficiency of these products is measured by yield of F starting from A. Further, the overall rate of conversion of A to F will partly depend upon the extent of coordination occurring between 5 enzymes. Because this is obvious, if more than one enzyme are participating in the formation of your final product F, then how much coordination is there in between those different enzymes? This will affect your rate of formation of final product. Now, for some metabolic pathways, certain enzymes are physically associated with one another to form multi-enzyme proteins. For example, fatty acid synthase complex and enzymes of tryptophan biosynthetic pathway. So these conditions actually point out that since there is existence of more than one enzyme in a multi-enzyme system, so those enzymes are physically associated, or we can say the physical association of various enzymes actually indicates the evolutionary existence of multi-enzyme system with time. So when we discuss about this topic of multi-enzyme systems, there are two common terminologies which are often used, and we should be very clear about this. So this is multi-enzyme complexes versus multi-enzyme polypeptides, and they are abbreviated as MEC and MEP. So let us see what are they. So multi-enzyme complexes. Multi-enzyme complexes are stable assemblies of more than one enzyme. more than one enzyme involved in sequential catalytic transformations. Remember, sequential catalytic transformation. For multi-enzyme complexes, it is very important to use this term sequential, because the whole idea of multi-enzyme complexes is based upon this fundamental concept that reaction they occur in a sequence and every step of the reaction is catalyzed by a particular enzyme. Whereas for multi-enzyme polypeptides, they are multiple catalytic domains found in a single polypeptide chain involved in catalytic transformation. So the basic difference between the two is where multi-enzyme complexes, they contain more than one candidate enzyme. On the other hand, your multi-enzyme polypeptides would be like the same polypeptide would be containing more than one catalytic domain, because ultimately it is your catalytic center, it is your catalytic domain which is responsible for the catalytic activity, which is responsible for the conversion of substrate into the product, what we call as catalytic transformation. For example, fatty acid synthase is a multi-enzyme protein that catalyzes fatty acid synthesis. It is not a single enzyme but a whole enzymatic system composed of two identical multifunctional polypeptides in which substrates are handed over from one domain to the next. So that was all about the basic idea, like how multi-enzyme system, the concept came into existence and what was the idea behind their evolution. Now let us have a look how do they evolve, or the phylogenetic distribution of multi-enzyme proteins. So let us start. So both MEC and MEP are more common and more complex in eukaryotes than prokaryotes. So more complex in eukaryotes than prokaryotes. This clearly indicates that your MEC and MEP have followed an evolutionary path, like the way your eukaryotes are more complex than your prokaryotes. So our enzymatic machinery have also evolved a parallel evolution mechanism, and this concept of multi-enzyme system came into picture. For instance, consider a prokaryotic cell such as E. coli. The cell of the cell has a very much smaller volume as compared to a typical eukaryotic cell. This we all know. And in eukaryotes, there is an intracellular compartmentalization due to various intracellular membranes. So the volume in which free diffusion of substrates and enzymes can occur is much greater than in prokaryotic cell. So this is very clear, since the size of your eukaryotic cell is relatively larger than your prokaryotic cell. So the volume into which your substrates can diffuse, or your intermediary substrates can diffuse, is much larger as compared to your prokaryotic cell. As a result, if intracellular concentration of substrates and catalytic centers are similar, now if you compare for a prokaryotic cell and for a eukaryotic cell, if there is a particular concentration of substrates and catalytic centers, then the diffusion time is more likely to be rate limiting in eukaryotes. How come? So let us first understand what is diffusion time. So diffusion time is nothing but it is the time to diffuse. Now in this context of multi-enzyme complexes or the sequential reactions, the time to diffuse would be the time taken by your intermediary substrates to diffuse or to travel within the cell from one catalytic center to the next. For instance, taking the same example of the reaction where A was getting converted into F, if A has converted into B, let me take an example. So if you have A, which was your starting substrate, and B, and suppose C, and then D is your final product. Now for conversion of A into D, you have two intermediary substrates, B and C, and let's suppose E1 is the enzyme or the catalytic center, E2 is the enzyme for B2C, and E3 is the enzyme for conversion of C to D. So let's suppose if your first reaction has taken place and A has converted into B via the action of E1, and if it is lying at this corner of the cell. Now in a cell, suppose this is your cell. Now in a cell, if B is lying over here and your E2 is lying somewhere here, so B2 would diffuse and reach to the catalytic center of E2, then only it will be converted into C. Likewise, C have to be accessed by your catalytic center E3, then only D will be formed. So how much time these intermediary substrates are taking to reach to their catalytic center is actually the diffusion time. And suppose if your volume is larger, then it is obvious that the diffusion time would be much larger if your catalytic centers or if your enzymes are not physically associated, or if they are lying apart, or if they are not lying in close proximity. I hope this point is clear. I'm again repeating, if intracellular concentration of substrates and catalytic centers are similar in prokaryotes and eukaryotes, then diffusion time is more likely to be rate limiting in eukaryotes. So suppose this is your prokaryotic cell and this is your eukaryotic cell. Now within the same cell, all your catalytic centers E1 to E3 are lying, whereas in eukaryote also E1 to E3 are lying. But since the volume is larger in case of eukaryotes, there are chances that your E1, E2 and E3 are not lying in the close proximity, and your intermediary substrate have to travel more in order to access their respective catalytic centers. So in that case, your reaction, your catalytic transformation, or the process of conversion of A to D, the process of conversion of substrate into product may take more time, or it may be slowed down. So that is why we are saying that diffusion time is more likely to be rate limiting. Now your rate of the reaction, rate of the conversion of A to D will totally depend upon the time taken by your intermediary substrates to travel or to access their respective, to travel from their one site to their next catalytic centers, or the time taken by the substrate to be accessed by the respective catalytic center is called the diffusion time, which may be longer in case of eukaryotes, and thus the reaction may go slow. So to cope up with such a kind of situation, evolution has resulted in multi-enzyme polypeptides. So as our cells have evolved from prokaryotes to eukaryotes, parallelly our enzymatic machinery is equally smarter, and evolution has resulted in a multi-enzyme polypeptides and more tightly associated multi-enzyme complexes. So the benefit of more tightly associated multi-enzyme complexes over here is that now they would be lying in close proximity of each other. Your all enzymes E1, E2, E3 and so on, they will be lying very close to each another, so that the product formed by the action of one of the enzyme would immediately be accessed by the next enzyme or the next catalytic center. So whatever loss could have happened due to the longer diffusion times is now minimized. Besides, genetic evidence also suggests that this is accompanied by clustering or even fusion of genes coding for these proteins. So that was all about the phylogenetic distribution of multi-enzyme proteins. Now let us start with the properties of multi-enzyme proteins. So basically, when we study about the properties of multi-enzyme proteins, we actually try to understand what are the advantages of enzymes being physically associated with one another rather than occurring as separate entities within cells. So let us see what's that. So the very first is the transit time. Now what this transit time is? So this transit time is the time required for a product of one enzyme to diffuse to the catalytic site of a second enzyme. I hope it is very clear. And since in our multi-enzyme complex systems or multi-enzyme proteins, the enzymes, they are physically associated with each other, so that particular time will be less in a physically associated multi-enzyme protein than those where there is no association. Further, this transit time also becomes rate limiting when the intermediate has a short half-life, or when an intermediate has a high relative molecular mass, then it would diffuse more slowly. So let us understand one by one. So if your intermediate has a short half-life, now again I will consider one reaction. Suppose again your A is the initial substrate and then your B, C are the intermediate substrate and D is the final product. Now suppose in a cell this particular reaction is taking place by the action of some enzyme like E1, E2 or E3. A is forming B by the action of E1, B is forming C by the action of E2 and finally C is forming D by the action of E3. Now suppose here B and C are your intermediary substrates. Now suppose your B is lying here in one corner of the cell where E1 was there and your E2 is lying here, and now your B has to travel from this point to this point. Your B has to travel from this point to this point. And also the half-life of B is very short. So short that the actual time taken by the B to cover this distance or the actual time taken by the B to be accessed by E2 is much longer than the half-life of B, which means by the time B covers its transit time or B reaches to the catalytic center of E2, before that only it starts degrading. So in that case, your transit time becomes a limiting factor and it affects your reaction rate. The other case is when an intermediate has a high relative molecular mass, then also it would diffuse more slowly. For example, carbamoyl phosphate probably has a short half-life in cytosol. Therefore, the association of enzyme carbamoyl phosphate synthase with aspartate carbamoyl transferase, which channels carbamoyl phosphate within the multi-enzyme protein, probably prevents its degradation. So, carbamoyl phosphate, which has a very short half-life in cytosol, so there could be a possibility that it may be degraded before it is accessed by its catalytic center. So, but by virtue of the association of the enzyme carbamoyl phosphate synthase with aspartate carbamoyl transferase, the two enzymes which are participating in that particular pathway, what they do, they channel carbamoyl phosphate within the multi-enzyme protein, probably prevents its degradation. That means they are somehow protecting the carbamoyl phosphate from getting degraded, and this they are doing by virtue of having this property of association. Another example of second situation could be pyruvate dehydrogenase system. Here, the second step in the reaction sequence involves the two enzyme-bound intermediates hydroxyethyl thiamine diphosphate pyruvate dehydrogenase and dihydrolipoamide acetyl transferase. So this is another example. Now the third property of multi-enzyme proteins is they have a reduced transient time also. Now what is this transient time? Let us understand this. So transient time is actually the time required to change from one steady state to another. Suppose in a system with two enzymes E1 and E2, and A is again the initial product and C is the final product, and the two participating enzymes are E1 and E2 and B is your intermediary substrates. So if there is a steady state in a cell, now if somehow the concentration of A is increased, then the time required to reach the new steady state will be much less in multi-enzyme proteins. Next property is channeling or compartmentalization. So this is very important property. So what is this? Once an intermediate has formed in a multi-enzyme protein, it is not generally available to be acted upon by other enzymes outside the complex. So this is the beauty of multi-enzyme protein. So what they have actually done is, I'll try to explain. Suppose this is again, I'm taking the same example, A, B and C, and let's suppose D. So there could be one case where A leads to the formation of B and then B would be converted into C. But how come when B will go and travel and reaches to the catalytic center of enzyme E2. Similarly, C will have to be accessed by the enzyme E3. Then only D will form. So in this case, there are high chances that as B would move out towards the catalytic center of E2, it may be acted upon by some other enzymes or it may be degraded. However, suppose if the situation is like this, that your enzymes has developed, and here is E1, that your enzymes has developed sub-internal channels or compartments through which your intermediary substrates can travel, and it is protected upon by other enzymes. So there are two benefits: one, it reduces their transit time, and second, it is actually protective for the intermediary substrates. So channeling would also reduce the loss of pathway flux by leakage or instability of free intermediates. So this is nothing just like that, just that your all the enzymes have actually form a channel within them through which your intermediary substrates can travel, like this. B will travel, then it is acted upon like that. Sorry, here is A will travel, A will be acted upon by E1, then B will be acted upon by E2, and like this. So such an arrangement has actually led to their protection of the multi-enzyme proteins, as well as they have reduced the loss of pathway flux or the loss due to the instability of these free intermediates. So this is a very important feature. Another important point is the multi-enzyme proteins, they act in a coordinated manner where one domain or polypeptide may exert an allosteric effect on the adjacent domain or polypeptide. For instance, in arome complex or arome enzyme, the substrate of the first enzyme is able to activate all 5 catalytic activities of multi-enzyme protein. This arome complex is a pentafunctional polypeptide which catalyzes steps of shikimate pathway. Another example is ribulose bisphosphate carboxylase oxygenase. This enzyme acts together with 4 other enzymes. So what are they? Phosphoribokinase, phosphoribose isomerase, phosphoglycerate kinase, and glyceraldehyde phosphate dehydrogenase. So your enzyme ribulose bisphosphate carboxylase oxygenase actually act in coordination with 4 other enzymes. What for? To catalyze 5 consecutive reactions of the Benson-Kelvin cycle. So this is another very important feature of multi-enzyme proteins. Now since your enzymatic machinery has more than one participant over here, E1, E2, E3, E4, E5, so it is always beneficial to work in a coordinated manner. And next property, there are changes in the kinetic parameters also. Obviously, when the system has evolved, then the multi-enzyme complex system, they will be having some change in their kinetic parameters also. For instance, their specific activity, which is expressed as kcat by km, is about 10-fold higher in the complex when compared with free enzymes. So now they have become like more robust. If their specific activity, they have become more specific. So this was all about the properties of multi-enzyme systems. Thank you. I hope you like this video. And for any queries, you may write into the comment section or you may contact me via mail. Thank you.